I'd like to discuss inflammation in a bit more detail. Inflammation involves three main stages. 1. Vasodilation and increased permeability of blood vessels. 2. Phagocyte migration and phagocytosis. And 3. Tissue repair. I'd like to start the discussion directing your attention to the top picture. Here we see a knife protruding through the integument system down to the dermis, which is filled with dense, irregular connective tissue. Mast cells and basophils release copious amounts of histamine and heparin in response to the injury. These chemicals result in dilation of the local blood vessels, leading to increased blood flow and increased blood vessel permeability. As a result, the area will become red, swollen, warm, and painful to the touch. As a result of increased blood flow, there will be more white blood cells, specifically monocytes and neutrophils, attracted to the injury site. The macrophages residing in the dermis when the injury occurred released cytokines. These are chemicals that attract other white blood cells to the injury site. The increased blood flow will also bring with it more clotting factors, and a clot will form in the tissue that stops the bleeding into the wound, but also walls off the wound to surrounding tissue, so any microbe introduced into the wound should be localized and prevented from spreading. When more white blood cells arrive at the injury site, T cells and antibodies also arrive, and these can provide specific defenses against any microbe that entered the wound. The macrophages and neutrophils will phagocytize any debris and also aid in the repair of the tissue. Now it is time to discuss the specific immune defenses. Lymphocytes include T and B cells. T cells, which give you cell-mediated immunity, and B cells, which gives you humoral immunity. These cells attack antigens in different ways, and I will discuss each one at length in the following slides. Together, these cells recognize specific antigens, but they are somewhat limited in that each lymphocyte can only recognize one particular antigen or epitope of an antigen. And when I think of this specificity, I think of a lock and key analogy. Perhaps you have used your house key to unlock your door. Perhaps you decided to do the empirical test and see if your house key was specific to your front door. You promptly walked to your neighbor's door and tried your key in their lock. Perhaps you discovered that your key does not open your neighbor's door. My house key will only unlock my front door, not my neighbor's. Let's pretend that the foreign antigens you encounter during your lifetime are represented as locks, and your T cells and B cells are keys. Each flu, virus, bacterial infection, or fungal infection you may ever have in your lifetime is represented as a new lock entering your body. The T cell and B cell that fought the infection you had last month fit into that lock, but the infection you have this month is a new lock, and thus a new key must be used to fight that infection. Obviously, this means we need millions of different types of B and T cells in our body. When one specific B or T cell encounters its antigen that it recognizes, it is activated and quickly creates copies of itself. Think of it as making duplicate keys. When the infection is over, many of the duplicated T or B cells die, while a few continue to live as memory cells. These memory cells will allow you to mount a quicker response and more aggressive response should you ever become infected with the same antigen in the future. This slide shows you how both B cells and T cells can form activated and memory cells. I'd like you to pay attention to the color of the nucleus for the cells depicted on this picture. The cell with a brown nucleus, which is shown here up at the top, represents a virgin cell. This means it is a B cell or T cell that has never been exposed to the antigen it can recognize and attack. When the virgin lymphocyte encounters its antigen, it replicates and makes clones of itself. Some of the clones will be active cells, those shown with a red nucleus, and fight the infection. Other cells with a yellow nucleus will remain dormant and serve as memory cells. When the infection has been conquered, the red nucleated activated cells will die, but the yellow nucleated memory cells will continue to live in the lymph tissues. Should the memory cells encounter the antigen again, they can quickly generate another army of activated cells. But the second response to the antigen will be much faster and more aggressive. Why? Well, with the first exposure, it was a single virgin cell that had to replicate to create clones of itself. With the second exposure, multiple memory cells were available for cloning. Sheer numbers dictate that the formation of activated cells will occur more rapidly and more abundantly. Using a graph to describe the quicker response to a second exposure to the same antigen, you'll notice on the x-axis the days elapsed after an antigen has been introduced into the body. On the y-axis, you'll notice the immune response. It is a gauge of intensity, and you'll notice there are no units associated with this axis. If antigen A is introduced into the body on day 5, you'll notice it takes 7 to 10 days before your immune response reaches its peak. If at a later date antigen A is reintroduced into your body, say because you catch the same cold again from your cousin or spouse, you'll notice your immune response takes a much shorter time to respond to that antigen. Additionally, it responds with more vigor. In other words, the immune response occurs faster and with more intensity. If, however, you are exposed to a different antigen, say antigen B, you'll notice that your immune response will again take 7 to 10 days upon first exposure to this antigen to mount an impressive response worthy to contain and eradicate antigen B. It is now time to talk more about cell-mediated immunity. Again, this is a specific branch of acquired or specific immunity that arises through T-cell action. There are 3 main T cells: cytotoxic T cells, helper T cells, and suppressor T cells. The helper T cell category is by far the most numerous. As their name implies, they help the immune system direct its response. Helper T cells are activated when their receptors bind to an antigen on the surface of an antigen-presenting cell. Helper T cells unite cell-mediated and humoral responses. When the helper T cell is activated, it will replicate and divide to create many clones of itself. It recruits more white blood cells into action. It can stimulate cytotoxic T cells, and it can stimulate B cells to mature into plasma cells that can then secrete antibodies. The cytotoxic T cell must have direct contact with its target cell. It is capable of destroying microorganisms and destroys virally infected cells in the body. For this reason, cytotoxic T cells are also called killer T cells. When a cytotoxic T cell binds to an antigen on a virally infected cell, the cytotoxic T cell releases hole-forming proteins on the surface of the infected cell. These proteins are called perforins, and they literally create holes in the infected cell's membrane. This causes the infected cell to begin a process called apoptosis. Apoptosis is a nice word to describe cellular suicide, or cell-mediated cell death. The infected cell starts to degrade its nuclear DNA and cellular proteins. When the process is over, the cell is dead and the virus cannot exist without it. Please understand the importance of this. Without your helper T cells, your B cells and cytotoxic T cells would be largely unresponsive to the infection in your body. If you could pretend to be HIV, the virus that causes AIDS, which of the T cells would you infect first and wipe out? If you guessed helper T cells, you would be correct. In fact, HIV infects helper T cells and depletes their population. All the while, the cytotoxic T cells are unresponsive to the infection going on. HIV is truly an insidious virus. Suppressor T cell function is not well understood, but we do know they are capable of suppressing the functions of cytotoxic and helper T cells. They prevent the cytotoxic T cells from generating excessive immune reactions that could destroy healthy surrounding tissue. Now it's time to talk about the other branch of acquired immunity, and that is humoral immunity. This immunity is conferred by the B cells in your body. When I say humoral immunity, it is because the B cells generate antibodies that stay in the blood. Blood is a humor, or fluid, and it is the antibodies that will latch onto a specific antigen. B cells do not engage in cell-to-cell combat like the T cells. B cells provide protection against foreign antigens that are in your circulation or outside of a cell. These antigens may arise from bacteria, viruses, or parasites, just to name a few. When the B cell becomes activated by an antigen-presenting cell, such as the macrophage, or activated by a helper T cell, the B cell will mature into a plasma cell. The plasma cell can make antibodies. I'll talk about antibody structure in a little while. Again, I want to emphasize that the antibody will only be able to recognize one specific antigen. That means you would need many different types of B cells, each capable of making only one type of antibody, to defend against the endless number of foreign antigens you may encounter. I want to remind you that when you come into contact with an antigen for a second or even third time, the memory cells will allow you to respond much more quickly and with greater quantity. In other words, if we're talking about a B cell response, they will create more antibodies more quickly. Memory cells are the basis for vaccine. Vaccines can be dead or weakened microbes, or just portions of them, like maybe just their epitopes, which are then introduced into the body and stimulates the immune system. Basically, vaccines cause you to create memory cells. If you should happen to come into contact with the antigen later in life, your memory cells are already in place and can rapidly respond with great quantities of antibodies, limiting how severe your illness will be. Here's a slide where I can stress to you the importance of understanding that B cells can only recognize one particular antigen. Shown at the top of the slide are 4 B cells with integral proteins shaped differently. You'll notice that only the one B cell that is square-shaped can recognize this square antigen. And once it does bind its antigen, that activates the B cell, and the B cell can actively replicate and divide and create more clones of itself. Additionally, that B cell will be stimulated into a plasma cell, or a mature B cell. And it is the plasma cell that can now generate antibodies that will specifically bind to that square-shaped antigen. Again, these cells are specific to one particular antigen. This is similar to the T cells that can only respond to one particular antigen. Clearly, you would need many different B cells and T cells in order to fight any invading antigen or microbe that you might encounter throughout your lifespan. Here is the basic structure of an antibody. It is a protein that is secreted into the plasma by a mature B cell called a plasma cell. Antibodies are also called immunoglobulins. You'll notice that they are a Y-shaped protein with 2 long peptide chains that are colored here as a dark brown and 2 short chains that are colored blue. The stem region determines the classification type of the antibody. There are 5 main classes. IG stands for immunoglobulin, and the classes are IgG, IgA, IgM, IgE, and IgD. I always remember this as, quote, gamed. I will discuss each of these and their functions later in this lecture. The arm regions that project outward from the stem consist of amino acids that allow the antibody to bind to one particular antigen. There are usually only 2 variable regions, one at the end of each arm, and it is here that the antibody gets its specificity in binding one particular epitope on an antigen. Larger antibodies such as IgM have multiple binding sites for a particular epitope. In the circle of this drawing, you'll see it is an expanded region of the variable end of the antibody, and you'll notice that it is in this variable region where a portion of the antigen can actually attach to the antibody. The antigen is depicted here as an orange-colored antigen with an extension called an epitope. An epitope, remember, is just a portion of a larger molecule called an antigen. An antigen is anything that can elicit an immune response. In other words, it's a foreign molecule that will cause your immune system to attack. There are 5 different classes of antibodies. Again, I like to remember the acronym GAMED. IgGs are the most abundant of all antibodies, and their functions include helping with opsonization, neutralizing toxins and viruses, and it's important that you know that they can cross the placenta and cause destruction of fetal red blood cells. This is called hemolytic disease of the newborn. You will have a lecture on hemolytic disease of the newborn later. IgAs are the antibodies that are found on the surface of your body. They are found on mucosal surfaces, sweat, tears, bronchial secretions, and even in your saliva. This little cartoon drawing shows you an IgM antibody. They are extremely large and can have 10 binding sites. In other words, 10 of the variable regions, each capable of binding the same antigen. They are the first antibody that is produced, and they can agglutinate antigens. In the middle picture of this antibody slide, you'll notice that the IgM antibody is the first to be created when your body is infected. IgG is the second class that is made. Levels of antibodies can be measured in the serum. In fact, we can take a patient's blood sample, load it into a gel, and separate the antibodies according to their size. That is shown in the bottom right picture of this slide. IgEs are the antibodies responsible for causing your allergic reactions. If you have severe allergies, IgEs are not your friend. They stimulate histamine release from basophils and mast cells, and I'll show you a step-by-step play later on in this PowerPoint presentation. IgEs are also important in the lysis of parasitic worms. Lastly, IgD. Well, we're not really 100% certain what they do, but we believe that they help activate B cells in some sort of fashion. Because of the bivalent nature of the antibodies and the multiple antigen sites on most invading agents or microbes, the antibodies can inactivate the invading agent in one of several ways. Through agglutination, in which multiple large particles with antigens on their surfaces, such as bacteria or red blood cells, are bound together into a clump held together by the antibodies that are attached to their surface antigens. This large clump can then precipitate. In other words, the antibody and antigen complex becomes so large that it is rendered insoluble and precipitates out of solution. Another way is through neutralization, in which the antibodies cover the toxic sites of the antigenic agent. Neutralization will be important when we talk about hemolytic disease in the newborn, during which a mother is Rh negative and pregnant with an Rh positive fetus. Next, lysis, in which some antibodies are capable of directly attacking membranes of cellular microbes and therefore cause them to rupture or lyse. Also, opsonization, which is what I've already covered earlier. Opsonization means that the antibodies that attach to the microbe can then attach to a receptor in a phagocytic cell, such as a macrophage, and this allows more potent phagocytosis. Additionally, the antibodies can lead to a cascade of events in the complement system. I will not go into the complement system as this is covered extensively in microbiology. Let's now focus on some pathological states of the immune system. Remember, the immune system must also have homeostasis. Too much or too little activity to antigens is not a healthy balance. Let's focus on hypersensitivities or allergies. Pretend that we are looking inside a small child who has just ingested some peanuts for the first time. Unbeknownst to the child, the peanuts are going to create a peanut allergy. Let's go step by step and appreciate how the peanut allergy will develop. The child has eaten a peanut, and the proteins of the peanut are antigenic to this particular child. That means the child's macrophage has docked to the peanut antigens, internalized it, and broke down the peanut. Some of the peanut antigens are now presented in the cell membrane of this antigen-presenting cell. You'll notice that here in the top left picture, where you see the antigenic peptide from the peanut presented in the macrophage. This macrophage, with its antigen presented, can now bind to a helper T cell and activate it. The activated helper T cell now releases many cytokines. Those cytokines can now activate cytotoxic T cells. Additionally, the helper T cell can activate a B cell, shown here in the top right-hand picture. The B cell has already bound the peanut antigen on its own and is also being stimulated by an activated helper T cell. Both of these routes can lead to a B cell that will rapidly replicate and divide to create clones. Some of these clones will remain as memory cells. Other clones will become mature B cells called plasma cells. These plasma cells in this example are secreting IgE antibodies. IgE antibodies bind to receptors on the mast cells. That is shown here with the red IgE antibody embedded in the receptor of a mast cell. Now, we have mast cells with the IgE antibodies embedded in their cell membrane. This same process could occur in a basophil as well. I want to remind you, now, if the child eats another peanut in the future, the peanut antigen can bind to the IgE complexes within the cell membrane of a mast cell or basophil. When the antigen binds, this stimulates the mast cells or basophils to release huge amounts of histamine granules, and this will lead to a severe allergic reaction, a lot of vasodilation, a lot of capillary permeability, and if enough granules are released, so much vasodilation and increased capillary permeability can occur that this can lead to anaphylactic shock, where the child's blood pressure drastically drops. Autoimmunity describes the loss of self-tolerance by the lymphocytes. Basically, the T cells and B cells now attack the body's own tissues. For some reason, and there are a few theories as to why, women are more afflicted with autoimmune disorders than men. Some of the autoimmune disorders we will learn or have already learned in this class include the list shown here. Graves' disease, you have already learned as an autoimmune disorder in which antibodies called TSI, thyroid-stimulating immunoglobulin, stimulate the thyroid inappropriately, and this leads to hyperthyroidism. Hashimoto's thyroiditis is another autoimmune disorder of the thyroid, but this time antibodies destroy the thyroid gland, and this leads to hypothyroidism. Another autoimmune disorder we will cover in the second unit is myasthenia gravis, and this is a disease where antibodies destroy the nicotinic acetylcholine receptor on the surface of skeletal muscle. As a consequence, the patient has progressive muscle weakness due to the loss of nervous stimulation of the muscle cells. You've also heard of one type of diabetes, also called insulin-dependent diabetes mellitus. In this case, antibodies destroy the beta cells of the islets of Langerhans found in the pancreas. This leads to the inability for insulin-dependent cells of the body to import glucose from the plasma. Multiple sclerosis occurs when antibodies attack the myelin sheath around neurons, and this leads to a decline in the speed of the action potential. MS will be discussed in more detail in Unit 2. Systemic lupus erythematosus is a chronic inflammatory disease that affects many systems of the body. The initial manifestation is a red rash around the nose and mouth and almost looks like a butterfly mask on the patient's face. In rheumatoid arthritis, it is a collagen disease and often ends up deforming the patient. This is when the antibodies chronically attack and destroy the collagen within the joints of the patient. The patient ends up with hands and feet severely flexed or curled inward. Extension of the joints is extremely difficult, if not impossible. This concludes your lecture on white blood cell physiology. Clearly this was a brief overview, and you should have received, or will soon receive, a more thorough knowledge of this system in microbiology.